Good morning. It's this time is really a great pleasure to welcome Professor Carlo Duarte for several reasons. Reason number one, because Carlos is in the family of Biogem, because he is not the first time. I don't know how many times he came here, but it's not the first time. But more important, many of our young researcher, young investigators, and the best judgment that a scientist can have is from the pupils, the fellows. Every time that a fellow came back from your institution, because they were quite enthusiastic. And many times I say, well, I've been. This is the best way I can introduce it. This means that you have, in addition to the let's say the important work that is on neuroscience, and I tell you why I'm very pleased about neuroscience. This means that if a fellow comes to you, you have a good human fiber. That is more important. You have good human fiber, a good human nature. That's, I think, the most important thing that I care. When I have to hire some of my fellow, I say he must be very intelligent, very active, but he should have also good character. If he doesn't have a good character, then it will pay the next, although it could be another price. So that's, I think, one of the best things that I can tell you. I'm very happy because Biogem, along the ages, along the time, is going to get some transformation. We have some labs that could be closed. Can be open in the near future. And the lab that we would like to open in the near future is a lab that in many interests will be the brain. We can say neurobiology, neurophysiology, whatever, but it should be the brain. Why? Because I think if you look to the years about science, if there is a part of science that has been extraordinary, impressive evolution, is the neuroscience. The reason why I say that because if we like to see an organ, it's very open. You open the chest, you open, like you can see. So far, we have never seen the brain because in order to have a brain, you have to go to school. That is something that is very well protected. But the last few years, our colleague neurologists and neurologists have opened the brain. We can see it. We are sleeping, we can see what we are eating, we can see part of the brain that flare up when you see, say, a wonderful woman, then part of it is when you are anxious. There's another part that's closed open, light up and light off. And also is very, very important because this means we are starting to understand how the brain functions. When we say the brain function, although I say all the time that the kidney is very, very important, but I must admit that the brain have something big different, because they can think, they can imagine. The other organ, they cannot do that. The imaging activity of the brain may depend from the function of the other organs. So we would like to open a neurobiology lab here. So you are very, very welcome. And if Lorella want to say something about Carlos, because I know that you know him better than me. It's the first time, but you are a very close friend of him, and particularly about his attitude. But according to the fellow, is very great.
Okay. First of all, I'm very glad today to introduce Professor Duarte, because Carlos has been a friend of, first of all, the University of Sagno, and Michele knows very well, and many students here, and there are some of the students that are here that had the opportunity and to be in his lab and actually at CNC in and the friendship and with not only with myself but with all the people in I can say the University of Sagno has been very important. With Carlos is very important because he helps so many students, and many of these students right now are researchers in his lab and are a researcher here. And basically, what I'm saying every time to Carlos is that going to Coimbra means usually we say that Erasmus changed life, but going to Coimbra changed completely a life in the life of these people. And many of these students right now are researchers, not only in Italy, but in Germany, in Belgium, in Portugal, and yes, in Portugal. And yeah, I like to say that the first student, Erasmus student that I had that was with the trainship, Miranda, right now is a researcher in his lab. And I also to say that many of the students, several students are married with Portuguese. Actually, we increased the number of it. Yes.
Inbreed.
Yeah. Yeah. There are a lot of kids, so we have also a new generation of neuroscientists, and so that's also important. Talking about, I mean, Carlos is I'd like to say that also is a great neuroscientist. Is one of the best, well-known scientists in the field of neurotrophin and so brain and the regulation of synaptic plasticity, and also in disease of the nervous system like stroke, like Alzheimer. And what can I say? It has been we I guess that we met the first time 15 years ago or even more. And in the last 15 years, I guess that more than 150 Coimbra. And so, and I think that, and also I would like to, and we have also the opportunity to start a double degree. Cinzia that participates to this program, Cinzia Mierelli. And as you know, maybe I can tell you that we have a double degree with the university. So our student, master student, they go there for 1 year and they get at the end a double degree, 1 degree from University of Sagno and 1 degree. And after this experience, the students, of course, they gain a CV and they gain a lot of experience. And actually, as I told you, they change completely their perspective of life and research and everything. And then they have many opportunities. So I'm very glad and to listen to. Okay, that's it.
So first of all, I'd like to thank you both for the very kind introduction. I'm really pleased to be back after COVID. And I should also say that we in Coimbra, we've benefited a lot from the visits of the students from Benevento who have been there for their internship, for their thesis. And because as we were discussing this morning, this is also very important for the local university, creates exchange of ideas and cultures, and this is part of Erasmus, which to me is one of the best and the most important projects of the European Union. So today I will be telling you about this work that we've been doing on BDNF in Italy. I don't have to tell you what neurotrophins are. And this is a photo that Pasqualino, a student from Benevento, took in the old part of the university. And I'll be very pleased to welcome you in Coimbra if one day you decide to visit our university and our city. So, so you don't need to be a neuroscientist to know that neurons are very polarized cells. And so this slide shows just a drawing of a neuron from the hippocampus, and these are cells that we grow in the lab. And this cell has a cell body that is located here, and it has a very long axon. And there are also these small dendrites that you see here. The fact that these cells are highly polarized created throughout evolution the need of or special needs in terms of delivering cell components to the different compartments. This is the case, for example, to the delivery of cargo to the distal part of the axon that you see here in the distal part of this neurites, but also the dendrites, even though they are shorter. Then, from the structural point of view, they're also highly specialized. And this slide just shows reconstruction of a dendrite of one of these neurons. And you can see that the dendrites is not completely flat. Instead, they have these protrusions that they have mushroom type of shape that which are regions the cell receives contacts from neighbor neurons, and these are called spines. And the contacts that the cell or the dendrite receives in these spines produce or induce the activity of the neurons. In contrast, in blue here, you see regions where the cell or the dendrite receives contacts from other neighbor neurons. Neighbor neurons, instead of enhancing the activity of the target cell, they inhibit the activity of these neurons. So just indeed, the synaptic contacts are made in different regions. But as I said, then the spines are very important for the signals that induce the activity, promote the activity of neurons. And these signals, they come from by that is called the glutamate. So in one of the, or some of the original electron microscopy studies that were performed to study the brain, it was possible or it was noticed that in this, near these spines, so you see here a spine, and near these spines, ribosomes. And as you know, the polyribosomes are regions of very active protein synthesis. And it was noticed that a large proportion of these polyribosome clusters, as this one here. And it was also observed that most of the polyribosomes, they would be found, they were not found elsewhere along the dendritic shaft. So this would mean that somehow first there is protein synthesis in this particular location. This particular location, if the protein synthesis does not occur randomly along dendrites, there should be a specific function for this local protein synthesis here. So, and here we're talking about coming back to the scheme that I showed you before. So these are the spines. Then protein synthesis would the spines here and here, but not along the dendritic shaft. So this was an observation that was forgotten for many years. The scientists then were attracted by this observation, and this was followed up by additional studies. And it is important because first, if there is protein synthesis located here, for example, this would allow to change rapidly the protein composition of the, and this would change, for example, the protein synthesis in this, the protein composition in this spine that was stimulated, while this one, for example, would not exhibit alterations in their proteome. Also, in terms of energy costs, it would be it would cost less energy to produce the proteins here that will be used in the activity of this particular synapse, rather than having the protein synthesized in the cell body and then shipped to the dendrites. And also, if the protein synthesis takes place in this particular location, then the alteration in the protein composition of the spine will be very rapid, very fast, in contrast with what we would see in case the proteins had to be synthesized in the cell body and then shipped to the synapse. So for proteins to be synthesized here near the synapse at the spine, as shown here in this particular diagram. Here, you need, of course, the messenger RNAs, and the messenger RNAs then they have to come from the cell body, and as you see in this diagram, then after being synthesized in the cell body, these messenger RNAs they will bind to proteins, and this is important because the interaction of the messenger these transcripts a lot more stable during their transport, and then they will be transported in these granules along the dendrites until reach an active synapse. And when they find an active synapse, then these transcripts will be recruited for protein synthesis and will change in the local protein composition. In case when the granules arrive here, if the synapse is not active, they continue traveling along the dendrites until eventually they will find another active synapse. So what I'll be talking to you today is about some of the signals that the synapse receives to change the local protein composition. RNAs are transported along the dendrites, and which is the functional relevance of having this local protein synthesis of the synapse. And it's this type of alterations or the local protein synthesis is particularly, for example, in what is called synaptic plasticity mechanisms, and this is something that has been characterized in the hippocampus, which is a for example, in formation of spatial memory. And on the left, you see here the synaptic organization of the hippocampus, and this here they were performed in the synapses that connect the axon of this yellow neuron with the green this region called CA1 region. And in this type of studies, what is found is that if you stimulate the yellow neurons at a low frequency, you get a consistent response by the postsynaptic, by the green neuron, and this response remains stable along the time. However, if you, for a short period of time, if you apply a high frequency stimulation to the yellow neurons, even though this high frequency stimulation occurs for a short period of time, then this will lead to a long lasting alteration in the connectivity between these two neurons. So you keep again, you come back to the old low frequency protocol of stimulation, then the response is potentiated. So this is called synaptic potentiation, and this is the type of alterations that occur at the level of the synapse that are thought to underlie learning and memory formation. For short-term, for the initial stage of the responses at this early stage, to a large extent, this potentiation comes from the fact that the receptors for the neurotransmitter can be there is a pool of receptors that is inside the cell, and these receptors can be inserted on the surface, on the plasma membrane of the synapse, so that there is an increase in the surface expression of the receptors for the neurotransmitter glutamate that is responsible for the communication. So having an increase in the surface expression of the receptors, then the postsynaptic cell will respond more to the same amount of glutamate that is released by the presynaptic cell. And of course, this is an alteration that does not require the synthesis of new components of this. It takes signaling mechanisms. So all the machinery is already available there. So this will then account for the fast response for the early stages of potentiation. At an early stage then of this potentiation, then additional mechanisms become involved, including synthesis. So one of the mediators of this potentiation is BDNF, and which is the neurotrophin that we've been studying for a long time and plays a very important role in the central nervous system, in contrast with the NGF that plays a more important role at the peripheral nervous system. And this is an experiment that, in vivo experiment that was performed by Gracien Lial, a former PhD student in the lab, that did similar to the one that I described in this previous slide, but in this case, this is the animals were kept. So this is an in vivo experiment. He was looking at synapses not in this in the CA1 region of the hippocampus, but in the axons that come from the cerebral cortex to the anti-gyrus, which is this other region of the hippocampus. But still, he found that high frequency stimulation leads to a potentiation of the synapse. And then when he used this chelates, which binds BDNF and prevents the interaction of BDNF with its receptors. So in this case, a decrease in the potentiation. So this shows that BDNF indeed is important for potentiation for the LFT particular protocol. And so what we've been studying is the mechanisms or the synaptic alterations that are induced by BDNF to this potentiation that in this case would account for this response. In particular, our working hypothesis was from the beginning was that stimulation of the receptors for BDNF, shown here, they would induce the delivery of receptors for the neurotransmitter, and this will increase the number of surface receptors present here, and by increasing the surface expression of receptors, then this cell will become more responsive to the glutamate that is released to the synapse. So in the first studies that we performed to address this question, we didn't have the tools to look specifically at the effects of BDNF on the synaptic expression of the receptors. So in these two papers that we published about 15 years ago, we just looked at the effects of BDNF on total expression of receptors at cell surface, and this includes the receptors here at the synapse, but also receptors that may be found here elsewhere on the membrane of the cell, away from. In both cases, we found that stimulation with BDNF of these hippocampal neurons increases the total surface expression of receptors for glutamate. And I should say that we looked at two different populations of receptors that are shown here in green and in yellow. And these are two types of receptors that bind the neurotransmitter glutamate, but they have different properties. They are both ion channels, have different functional properties. One is called the AMPA receptors, and the other one NMDA receptors. And so here you see that both indeed increases the total surface expression, the total amount of receptors present at the cell surface, including the synaptic receptors and the receptors away from the synapse, both for the AMPA and the NMDA receptors. So both populations of receptors for glutamate. So it was more recently than that we set up the tools to study the effects of BDNF on the expression of receptors at the level of the synapse. And a set of studies that I'm going to present today, I'll be focusing on the NMDA receptors. So this is the green receptors that, and actually for a long time it was thought that the AMPA receptors would be very dynamic, and the abundance of these receptors at the level of the synapse would change depending on the activity of the neurons. we now know that also the NMDA receptors, they are quite dynamic. Perhaps they are not as dynamic as the AMPA receptors, but their abundance at synapse can also change depending on the activity of the cells. So in this case, what Pasqualino De Luca did was also a student, first student of the double degree program between Unisanio and the University of Coimbra. And then he followed up and did the PhD also in our PhD program, and he's now a postdoc. And so he set up these electrophysiology experiments in the lab to look at activity of NMDA receptors for glutamate. And in this case, he was looking at response to glutamate that is released spontaneously by presynaptic cells. So typically we hear about neurotransmitters, and then this leads to the calcium-dependent release of neurotransmitters. But there may also be some spontaneous release of neurotransmitters. And in this case, what is looking at is the activity of this population of receptors, NMDA receptors, that are activated when the presynaptic neurons release spontaneously glutamate. And this gives this type of recordings with intracellular pipettes. And so this is the type of event that is recorded whenever there is release of glutamate by the presynaptic cell. So the frequency of these events will tell us whether the presynaptic cell is releasing neurotransmitter more often or less often, whereas the amplitude of these events will tell us whether there are more postsynaptic level, so that the postsynaptic cell will respond more to the same amount of neurotransmitter. So what you see here is that BDNF stimulation indeed increased the postsynaptic response, indicating that there is, or suggesting that there is an increased amount of receptors at the postsynaptic cell. And when they blocked these receptors, then you no longer could see an effect of BDNF. So this shows that indeed BDNF increases the synaptic abundance of the receptors. And here just illustrating the structure of these receptors. They are formed, as you see here, by 4 subunits that they cross the membrane, and they limit the channel at the center that allow the ions to flow across the membrane of the cell. So what we're looking at here, so these NMDA receptors, they actually found in the hippocampus, they actually may come. And what we're looking here specifically at receptors that contain this type of composition, the GluN2B receptors, and which also for many years were thought to be not to be very abundant at the synapse. We know now that they can actually be present in significant amounts. As you see from here, then the abundance of these receptors at the synapse can also be changed depending on the pattern of stimulation of the cell. Can also then in additional studies that Pasqualino did, he tried to understand the mechanisms whereby BDNF increases the abundance of synaptic receptors, and to this to address this question, he did studies labeling single receptors with antibodies, as you see here. In this case, an antibody against the subunits of the receptors that are bound to quantum dots, which are highly, very bright fluorescent particles. They are so bright that you can actually microscope in imaging experiments. You can just follow the dynamics of a single receptor on the surface of a living cell. And here you see in green the dendrite of a cell, and these red particles here are single receptors that are labeled with this technique. And in this case here, I don't know if you can see here, for example, there is a spine that comes from the dendritic shaft. there was this video that we could see before and we cannot see now. Now I can see. So this is just to tell you, to show you that these receptors, they are not immobile on the membrane, they are quite dynamic. And then after recording the movement of the single receptors, then at the end we can just analyze the dynamics of the receptors and study the receptors that are found at the synapse and the ones that are away from the synapse and see how dynamic they are and whether their dynamics when the cells are stimulated with BDNF. And this is what you see in this. Um, so here, for example, see under control conditions, and so this is a quantification of the area that is covered by the under control conditions in the absence of BDNF. And here in red, you see the area that is covered by the receptors on average. The synaptic receptors has been stimulated with BDNF. And you see that the receptor after BDNF stimulation, the receptors tend to be within a very narrow area when compared with the with the control conditions in the absence of BDNF. Also, when the the rate of movement, how fast the speed of movement of the receptors, and the control conditions and simulation after simulation, we see that BDNF stimulation makes the receptors moving slower when compared with the control conditions. Shows that indeed after BDNF stimulation, the cells or the receptors become less mobile. And this is in agreement, indicating that there is an increased amount of receptors at the synapse, just because, most likely because they are less. And here to further address the mechanism whereby BDNF stimulation increases the amount of the activity of synaptic receptors. Again, Pasqualino went back to the electrophysiology experiments and the response mediated by these receptors after BDNF stimulation. And when he did the same experiment, you see that BDNF no longer had an effect on the response. So this means that protein synthesis is indeed important to increase the activity of synaptic receptors. This can also be illustrated in this immunocytochemistry experiment, where we labeled in the cultured hippocampal neurons once again, where in these experiments we used an antibody against the NMDA receptors in cells that were not fixed and not permeabilized. So under these conditions, then the antibody, as you see here, it binds only to the receptors that are surface. It doesn't bind to intracellular receptors just because the membrane is not permeabilized. And then later on, we fix the cells. The cells are permeabilized, and then they are incubated with an antibody against the vesicular transporter. So this antibody will bind to the presynaptic region, and a third antibody that binds to a postsynaptic protein that will label the postsynaptic region. And in these experiments, then what we do is to colocalize the presence of the labeling of the receptor with the labeling of the presynaptic marker and the postsynaptic marker. And this type of labeling in immunocytochemistry experiments will tell us about the presence of synaptic receptors. And this is the type of labeling. So you get the punctate distribution that is in accordance with the clustering of the receptors at the synapse. And then, as I said, then we did the colocalization with this protein here, which is present here. And also, so this is the vesicular transporter that labels the presynaptic region. So you see again that BDNF stimulation increases the synaptic labeling of receptors. And this is also blocked by a protein synthesis inhibitor. Again, showing that the abundance of receptors at the synapse, and this depends on protein synthesis. This actually reminds of studies that were performed a while ago in the lab of Erin Schumann, where she looked at they looked at the effects of BDNF on the synaptic response in the hippocampal synapses in this region, as I showed you before. So BDNF stimulation potentiated this response, and this was blocked by protein synthesis. So again, showing that protein synthesis is really important for the effects of BDNF at the synapse. So the question then we asked is, so BDNF stimulation somehow leads to potentiation of the synapse due to protein synthesis. How is it that the transcripts arrive here to be used for protein synthesis for synaptic potentiation? And for that, then we relied on studies that we had done long ago. A proteomic study doing 2D gels followed by protein mass spectrometry. At the time, the goal of this project was completely different. So we were interested in neuroprotection by BDNF. But when we went back to these studies, we found the population of hippocampal neurons changed the intensity of two particular spots that are shown here that were identified as this protein here called heterogeneous nuclear ribonucleoprotein K. So this is a protein with a very long name, and in the lab we just call it RNPK to make it simple. And and since this is an RNA binding protein, so even though this is a protein that had been poorly studied in the nervous system, had been mainly studied in the context of cancer, and but this is a protein that binds RNAs and also plays a role, for example, as you see here, in cell signaling, in the regulation of translation and stabilization of RNAs, shuttling of RNAs, transcription regulation, so it plays numerous roles. But we just hypothesized that perhaps this protein could play a role in the regulation of protein synthesis here at the synapse just by binding transcripts here at the soma and transporting these messenger RNAs along the dendrites. And in fact, when he did these Western blot experiments with an antibody against the phosphorylated form of the protein, we saw that BDNF stimulation increases the phosphorylation of RNPK. So this also suggested us that somehow BDNF stimulation could regulate the function of this protein. And importantly, when we went back, when Pasqualino went back to his electrophysiology experiments to study the effect, the response, or the activity of the synaptic receptors under control conditions and after stimulation with BDNF. As I showed before, BDNF stimulation increases the amplitude of the response in accordance with the idea that there is an increase in the surface expression of the receptors at the synapse. When he knocked down the RNPK protein, then BDNF was no longer able to increase the synaptic response. So this was very interesting because it suggested that somehow this RNA binding protein may be important to carry the transcripts along the dendrites towards the synapse, and these transcripts then would be important and used for protein synthesis for synaptic potentiation. And the question we asked then was which are the transcripts that bind to this RNA binding protein. And this is the result of a study in which we immunoprecipitated the RNPK protein from cultured hippocampal neurons. And then we did microarray analysis to identify the transcripts that were bound to or that were co-immunoprecipitated with this protein. And we were really surprised that about 16,000 transcripts were identified. Perhaps not all of them bind directly to this particular protein. Some of them may bind to other RNA binding protein that interacts with the RNPK protein. But what was important to find was that Many of these transcripts, for example, they code for proteins that are important for synaptic structure and function, regulation of synaptic plasticity, learning, dendrite developments, so all functions that are important at the synapse. So again, this suggests that somehow this RNA binding protein may be important to deliver transcripts to the synapse to be used for local protein synthesis. Then the question we asked next was what happens to the interaction of these transcripts with RNPK protein when the cells are stimulated with the BDNF. So we took two pools of cells, one pool that was maintained under control conditions and the other one that was stimulated with BDNF. Then we immunoprecipitated the RNPK protein, and then we analyzed the transcripts that were bound, that were co-immunoprecipitated in the two pools of cells. So the transcripts that were co-immunoprecipitated under control conditions, and those that were co-immunoprecipitated in cells stimulated with the BDNF. And so this graph here shows the results, plots the results obtained. So on the left, you see each one of these dots are transcripts that show a decreased interaction whenever the cells are stimulated with the BDNF. And on the right, you see transcripts that show an increased interaction after stimulation with BDNF. So you see, I can notice that there are no transcripts showing a significant increase in interaction. What we saw was a decrease in the interaction of the transcripts with the RNA binding protein after BDNF stimulation. So, and this is again suggests that when the cell is stimulated with the BDNF, then there is a pool of transcripts that are bound to the RNPK protein that become dissociated, so they will become available to be used for protein synthesis. And out of these 16,000 transcripts that we had identified, about 9,000 show the decreased interaction after stimulation with BDNF. And when analyzed by their function, they also show functions that are important in terms of neuronal regulation. So then, of course, we had all these transcripts, and we had to do something with it. And then our next goal was to try to find a protein that would be coded by one of these transcripts that could then play an important role in terms of synaptic regulation. And one of the, well, we focused on this kinase called PYK2, and this is a cytosolic tyrosine kinase. So it's not does not interact with the membrane, and is known to be activated in response to neuronal activity. And this graph shows the alteration in the interaction of the PYK2 messenger RNA with the RNA binding protein whenever the cells are stimulated with the BDNF. So level 1 shows the interaction under control conditions. And what you see here is that when the cells are stimulated with BDNF, there is a decrease in the amount of PYK2 messenger RNA that interacts with RNA binding protein. So suggesting that BDNF stimulation can lead to the local synthesis of this kinase. And so this is a kinase that can be activated in response to neuronal activity. So it can be autophosphorylated to be activated, and then can regulate, for example, SRC. And SRC has many targets at the level of the synapse. And it was interesting, and that was actually one of the reasons why we focused on this kinase. That's because there were previous studies showing that the PYK2 could actually regulate the activity of the glutamate receptors. And so we thought that perhaps this kinase could be an important mediator in the response induced by BDNF to enhance the levels of the receptor at the level of the synapse. So this was to further investigate this issue. we picked up synaptoneurosomes, and synaptoneurosomes are an old preparation which basically contains the presynaptic region and the postsynaptic region together. And this is a functional structure and allows us to study the local alterations in protein synthesis whenever the synapse is stimulated. So what we did was we isolated synaptoneurosomes from the hippocampus. Then we kept a pool of synaptoneurosomes under controlled conditions, and the other pool was stimulated with a BDNF. And then we prepared lysates of the synaptoneurosomes, and then through a gradient, then we can isolate the polysomes, which are involved in protein synthesis, monosomes, and pre-monosomes. And this gradient can be eluted. And here we have the fraction corresponding to the polyribosome fraction that was on the bottom of the gradient. And then after extraction of these fractions, then you can do real-time PCR and analyze for the presence of different transcripts in these fractions. And what you see here is that indeed the PYK2 messenger RNA is significantly increased in the polyribosome fraction in synaptoneurosomes stimulated with the BDNF, indicating that this protein can be synthesized locally in response to the stimulation with the neurotrophin. I think I will just skip some slides. And then we did additional experiments where we found that indeed BDNF stimulation can induce local synthesis of the PYK2 kinase. And the question then we asked was whether the RNA binding protein was important in the synthesis, local synthesis of the kinase. So for that, then we took again the hippocampal neurons in culture, and we kept them under controlled conditions. Stimulation with BDNF increases the levels of the kinase, and when we knocked down the RNA binding protein, then there was no effect, no increased synthesis of the PYK, no increased levels of the PYK2 protein. So again, showing that BDNF stimulation, or in this case, the RNA binding protein, RNPK is an important mediator in the local synthesis of the kinase. Then finally, the other question then we asked just to conclude the story was to check whether the PYK2 was indeed important on the effects of BDNF on the synaptic expression of the receptors. And these are experiments similar to the ones, immunocytochemistry experiments similar to the ones I showed you before. So BDNF stimulation increases the synaptic expression of the receptors, but when we knock down the PYK2 kinase with two different shRNAs, then there was no effect of BDNF, showing again that PYK2 is indeed an important mediator in the response to BDNF in terms of the increased expression of NMDA receptors. So what we think now, or our goal now, is to further investigate this issue to show a role of the surface or synaptic expression of these NMDA receptors in the LTP process. And this is the result of experiments that we have performed together with a colleague from the University of Lisbon. And so these are synaptic potentiation experiments similar to the ones I described to you in the beginning. So high frequency stimulation leads to the potentiation of the synapses in the hippocampus. If you stimulate this, do the same protocol in the presence of BDNF, then there is an increased response. And in this particular experiment, then we blocked the NMDA receptors that I've been talking about, the ones that have this particular subunit composition. And then you see that under these conditions, then there is no potentiation by BDNF anymore. So showing that indeed the stimulation or the effect of BDNF on the surface expression, on the synaptic expression of these receptors are important for the potentiation of the synapses. This is under more physiological conditions, under normal conditions. And we believe that this type of effect, the effects of BDNF on the surface synaptic expression of the receptors may also be important in the context, for example, in epilepsy, when it's known that there is an increased levels of BDNF at the level of the synapse, and we believe that under these conditions there may be an increase, an excessive increase in the synaptic expression of the glutamate receptors, and this may then lead to an excessive activity of the excitatory synapses that is characteristic of seizures. So, so just in summary, what I've told you about yesterday is that indeed local protein synthesis is important in the regulation of the activity of the synapse. That BDNF that activates the TrkB receptors shown here as the important mediator. BDNF acts through by promoting the dissociation of transcripts that bind to the RNPK RNA binding protein. And PYK2 is one of the targets that gets synthesized locally. And this protein is important, or this kinase is important to upregulate the surface expression of these receptors for glutamates that are shown here in green, that are named NMDA receptors. So, so this is a recent photo of the group that we took a couple of weeks ago. And so these are some of the people that did work, in particular Pasqualino De Luca, who, as I said, was a student of the University of Sanu, together with Miranda Mele, that was also one of the first students from the Unisanu in Coimbra. And most of the funding came from FCT, which is the National Funding Agency. So thank you a lot for your attention, and I'm open for questions.
Thank you very much, Carlos, for this exciting, quite new family. I mean, from me, for most of our fellow here. To understand that it's a quite complicated issues, but very, very important. We are speaking about synapses. It's one of the measuring function of the brain. It's complicated, but I think how we have to, some curiosity. I don't know if Lorraine would like to start as an expert, or I can go ahead. Okay, I have two questions now. The first one is about all these synaptic proteins that are involved in the transmission. How much is the control intracellular, and how much is extracellular? In other words, all these proteins that are contributed to the synaptic interaction is controlled only from the cellular space, or also from factors that are extracellular, mean dependent from other organs.
From other organs.
Yeah, sure. Because I know this is quite complicated, but that's one of the topics we would like to see also if how can contribute or can modulate, or can give them up and down that these synapses messages.
Yeah,
that's good.
So I'd say that I think most of what
if you can speak by
yes. So I'd say that most so you're talking about intracellular versus extracellular, and from the extracellular, what is local and what comes from the outside.
Exactly.
That's so in terms of for the BDNF, for example, that is the signal that triggers this local protein synthesis that I've been talking about. So actually, so the BDNF that causes this local protein synthesis, BDNF is stored in vesicles, at least in the extracellular space, in the postsynaptic region. And so then depending on the groups, some people tell you that most of it will come from the postsynaptic cell. Others will tell you that there is also a significant component, or most of it come from the presynaptic region. But but it's stored in vesicles, and then in response to neuronal activity, these vesicles fuse with the membrane, in a way similar to the exocytosis of the neurotransmitters, and this releases these molecules, and then this BDNF then will target the receptors. These receptors can be presynaptic, and if they are presynaptic, then they can, for example, enhance the release of glutamate, and by enhancing the release of glutamate, then there'll be more neurotransmitters, so there will be an increased response by the postsynaptic cell. As I discussed, then this BDNF can also target postsynaptic receptors to induce protein synthesis and to potentiate the synapse at the postsynaptic level. So this is BDNF itself. But BDNF is synthesized as a pro-neurotrophin. There is called a pro-BDNF. And the pro-BDNF can be cleaved to give rise to BDNF inside the cell. In this case, we have the release of the BDNF, but can also be released as pro-BDNF. And when it's released pro-BDNF, then its activity on these receptors that I've been talking about is dependent on the cleavage of the pro-BDNF molecule to give rise to BDNF. And this cleavage is mediated by extracellular proteases. So this is one example of an extracellular event that is important to control the synaptic response. And in case the molecule is cleaved, then it activates a certain population of receptors that are named P7, the TrkB receptors, and which are the receptors that mediate these responses that I discussed. But in case it's not cleaved, then there is a second population of receptors that I didn't talk about. They're called P75 neurotrophin receptors that are activated by the pro-neurotrophin, by pro-BDNF. And it's interesting to notice that the responses that are obtained in that case, they are completely the opposite. So for the mature form of the neurotrophin, then the type of response that we get is the one I described, in the sense that they potentiate the response, the synaptic activity. For the pro-neurotrophin, since it targets a different pool of receptors, then instead of making the synapses stronger, then it makes the synapses weaker. So if the molecule is not cleaved, then the synapses actually may become weaker. And so this is in terms of local mediators. But in terms of signals that come from the outside, what comes to my mind first is the work of a colleague of mine that will show that the ghrelin that is very much associated with the food intake, that also regulates synaptic activity. And so can also, what I described here in terms of regulation of synaptic receptors, for example, and dynamics of the synaptic receptors, ghrelin has this type of effect also.
I think that it was already a complicated story, become more complicated if the other some more. Any question from?
Yes.
Okay.
Yeah. Any particular mouse models study?
So for this type of studies, no, we've been relying mainly on the shRNAs that we use to knock down specific proteins, and because you cannot, for example, if you knock down BDNF or the TrkB receptors, these molecules are so important for the cell, for neurons, that you cannot keep these animals. So what you can keep the heterozygous animals to do some in vivo experiments, but you cannot keep knockouts because they are not viable.
Okay, just a brief result. Thank you for this introduction on this very specific, very accurate. I just a curiosity. From the clinical, or are there any disease associated with the dysfunction of this signaling? So is there any age-associated disease or rare disease associated with dysfunction?
So, for example, I recently came across with studies where they found alterations, variations in the RNPK protein. And they found that these patients, they are characterized by mental retardation. And I think in this particular context, it makes a lot of sense. And these are very recent studies, 3, 4, 5 years old studies. And I think this is something that we should pursue. And also, there are some variants of the BDNF that can be expressed that can also cause some cognitive alterations. And
so there are some specific alterations or mutations that are associated with an aberrant expression or behavior of BDNF. Right?
Yes, and also, for example, as I was saying, then in epilepsy, it's thought that the initial, for example, can be started at an early stage when a set of neurons show an increased activity, and this increased activity then will lead to an increased local production of BDNF. And then this BDNF, then the idea is that this BDNF then will be released, will potentiate the synapse, perhaps based on these studies. I think that at least part of this potentiation will be mediated by the fact that there will be an increase in the surface expression of glutamate receptors. And so if there are more receptors, then the synapses will become more active. And then if they are more active, then there is more production of BDNF, more release of BDNF, and then this makes a loop that will then just leads to the seizure activity. And this is also an example that I think that's something that we are pursuing right now. And because cannot just block the BDNF receptors because they are so important for the cells. And so if you want also to act in this loop from the therapeutic point of view, you have to find out the mechanisms that act downstream in order to block them. Otherwise, then if you block BDNF, since it's important for cognition, for example, then you have cognitive problems also. Also, it's important for neuronal survival. And it's important for differentiated neurons to keep them alive. You need a continuous supply of BDNF. So if you block the receptors for BDNF, then you'll kill the cells and the patient.
Question? Antonio? Voice.
Is it possible to connect with the Parkinson's disease or the potential symptom in some neurological disease?
I think so there are studies more on Alzheimer's disease and in Parkinson's disease. I think also, for example, in Huntington's disease, that's a disease where the alterations in the trafficking and delivering of BDNF has been largely correlated with neurodegeneration, for example. That's a very it's a typical example that is commonly mentioned of a neurodegenerative disorder, related in this case with an impairment of and in Huntington's disease, there is this antigen-associated protein that is a scaffold protein that is very important for the trafficking of the vesicles that carry BDNF to the synapse. And the mutant protein then is not able to mediate the transport of these vesicles, of this cargo, and so this leads to a decreased downregulation in the delivery of BDNF to the synapse, and then this leads to neurodegeneration. It's probably the best example I can give you.
Question? Sure.
Thank you for the great talk. And I was wondering, did you also check a reducing reuptake of the glutamate in the synaptic space also, or just an increase of the receptors?
So in that field, the only experiments that we did was more on the really exocytotic release of glutamate. And for that, what we did was we isolated synaptosomes, and this is something that Lorella knows very well. And so we isolated synaptosomes, and with this preparation, then these are vesicles that contain all the components that we find at the axon terminal, including synaptic vesicles, mitochondria, and they keep a membrane potential. So you can stimulate them and look at the release of neurotransmitters, including glutamate. And what we did was to stimulate control synaptosomes and synaptosomes that were incubated with BDNF. And what we found was that we and others was that BDNF stimulation increases the release of neurotransmitter, so in release of glutamate. So this means that in addition to the postsynaptic responses, there are also receptors for BDNF at the presynaptic level that potentiate the release of neurotransmitter, potentiate the release of glutamate. So under this condition, there will be more glutamate at the synapse. There will be more a stronger response by the postsynaptic cell. So there are pre and postsynaptic effects. Yes.
I have a question about thanks a lot for this seminar. My question is about the presence of BDNF along the evolutionary tree of animals. And where does it appear and how is it positioned along the evolution? And the back part of this question is if is if it would be possible to find some relatively simplified animal models to investigate biology of BDNF, of this molecule.
Yeah, that's a very interesting question, but I have no idea how to answer. And but that's actually I think that's something that I should go back and study, because this is really interesting and important question. The only thing I know, for example, that there is NGF, BDNF, NT3, and NT4/5, and these are the neurotrophins that we find typically in mammalians. But there is also neurotrophin 6 and 7 that we find in fish, but we don't find, for example, in mammalians. So I know if there was some ancestor neurotrophins that then give rise to the neurotrophins that we find, for example, in mammalians. This may be an hypothesis, but that's something that we should go back and study. Thank you for your question.
Arthur? Council, in one of your slides you mentioned about rapamycin. Yes, you know, rapamycin is a well-known drug that has been used for in several diseases because it's very, very strong immunosuppressor. And I think you quoted the fact that rapamycin may suppress the synaptic activity of those.
So it blocks some of the effects of BDNF, because so the TrkB receptors, they signal, so these are tyrosine kinase receptors, and so they when the receptors are activated, they dimerize, and then they transphosphorylate each other on tyrosine, and then this leads to the activation of three main signaling pathways: the PLC gamma pathway, the ERK pathway, and the PI3 kinase pathway.
They activate or they disactivate?
They activate.
Activate. So it's anything to do. So the rapamycin can be involved in some, let's say, loss of function of the brain, because we use a lot in clinical practice.
So actually, so there were some experimental studies on some models of epilepsy that they used rapamycin, and it was actually protective in terms of acting against the development of seizures. And so it was thought for some time that in some cases rapamycin could actually be.
How you can foresee if you use rapamycin in patient, cognitive capacity of the brain should be decreased or increased? Sorry for the question, but if it's very
in terms of the, yeah, it's I don't think the activation of the AKT pathway induced by TrkB receptors is not the main player in terms of the effects of BDNF in synaptic plasticity. So from that point of view, that is more related in terms of regulation of cell survival, for example. And so I don't think this will be this will have from that point of view, just considering the response to BDNF stimulation, I don't think rapamycin will have a significant impact on the effects of BDNF on cognition.
So that's Lorena. It's your turn.
Yeah, I've discussed this so many times.
I know that you know that well all these words.
And
I think we have collaborated. But
Lorella has read so many theses coming from Coimbra on
you
this
can
topic.
conclude
that
it.
She's also an expert on the topic.
The question is more related to the ionic mechanism of the so instead you look at the calcium or
So in terms of calcium regulation, since BDNF, as I said, then activates the PLC gamma pathway, so so it ends up leading to the recruitment of calcium that is stored in synaptic vesicles through an IP3 dependence mechanism. That is the main target. But then the regulation, activation of the TRAC receptors also leads to the regulation of these the TRAPC channels that are non-specific ion channels that can also regulate directly the plasma membrane permeability from that mechanism.
so the trigger could be the activation of the metabotropic glutamate receptor, calcium, or it's more related to alpha or
So in this case,
the postsynaptic.
So in this case, there will be direct activation of the PLC gamma by the TRAC-B receptor without the need of activation of a metabotropic receptor. Because it will be a different PLC phospholipase C isoform, because this one, the PLC gamma, is the one that is directly involved in the response induced by the neurotrophin receptor. So this PLC isoform is different from the one that is activated by the metabotropic receptors. But at the end, then in both cases, we have recruitment of mobilization of calcium from IP3 sensitive stores.
And if anyone has a question, I would like to thank Jack Allen for his excellent